we've established that a key question is how genes influence behavior. But let's peel back another layer. Forget the simple blueprint idea for a second. Think of your genome as a bustling city with countless traffic signals. These are our transcription factors, or TFs. TF might be the signal for Main Street, turning on genes 1, 3, 5, 7, and 9. TFB controls Wall Street, activating genes 1, 2, 5, and 6. And TFC manages Park Avenue, dealing with genes 1, 5, and 10. Notice gene 1? It's got multiple addresses, multiple promoters, each responding to different TFs. This means one gene can be part of totally different regulatory networks, depending on which TF shows up. And it works the other way too. One TF, like NF-kappa B when cells get injured, can flip on an entire network of inflammation genes. Now imagine a glitch, a mutation in the address for gene 3 that only TFA knows, making it respond to TFB instead. Suddenly, gene 3 gets pulled into the Wall Street network. Same outcome if the TF protein itself mutates and starts binding to the wrong address. The city's traffic flow changes dramatically based on these tiny mutations. And just how complex is this genetic city? Hold on to your hats. We're talking about roughly 500 different types of traffic signals, TFs, in the human genome. But the map, it's got 4 million potential stoplights, and the average cell, to do its job, only needs to use about 200,000 of them. That's like having a massive, multi-city transit system where only a fraction of all possible routes are actually used by any single commuter. It's mind-bogglingly complex. This isn't just about having the parts. It's about the intricate combinations and regulations needed to build a functioning organism. It tells you right away that gene expression isn't a simple on-off switch, but a highly orchestrated combinatorial dance. Okay, let's talk about something even more subtle, epigenetics. Remember how we said the environment freezes genetic switches? Epigenetics is the molecular mechanism behind that. Imagine the DNA strand as a highway. Normally, transcription factors, TFs, are like cars looking for specific exits, promoters. But an epigenetic change is like someone parking a giant truck directly across the entrance ramp, or attaching a big sign saying no entry to the ramp structure itself. An environmental event triggers this chemical modification. Maybe a methyl group latches onto the promoter region or the surrounding proteins. The result? The TFs can't get through, can't bind properly, and the gene downstream is silenced, effectively turned off. Think about those prairie voles again. Their first mating experience doesn't just create a memory, it physically alters the regulation of their oxytocin and vasopressin receptors in the brain's reward center, the nucleus accumbens. This epigenetic tweak helps lock in their pair bonding behavior. These aren't just temporary glitches, they can stick around for a lifetime. Now here's where it gets really interesting, and frankly, a bit controversial. For decades, the dogma was that all these epigenetic marks get wiped clean during the formation of eggs and sperm, ensuring a fresh start for each generation. But guess what? Turns out that's not always true. Some marks survive the journey. There are now clear examples, like making male mice diabetic and then watching their offspring develop diabetes too, solely because of inherited epigenetic changes in the sperm. This brings us face to face with Jean-Baptiste Lamarck, the guy everyone mocked for suggesting giraffes stretched their necks longer and passed longer necks to their babies. Well, in this very specific, narrow sense, Lamarck was right. These neo-Lamarckian inheritance mechanisms show that acquired traits, or at least their environmental triggers captured epigenetically, can indeed be passed down. It means the environment doesn't just affect us, it can affect our kids, and maybe even our grandkids, through these molecular imprints. All right, let's shatter another cherished DNA dogma. You learned one gene, one protein, right? Cute, but mostly wrong. Most genes are actually modular, like a kit with separate pieces. Imagine a gene for building a chair. Instead of one long instruction manual, you have separate sections for the legs, the backrest, the seat, and the arms. These coding sections are called exons. But between them, there are non-coding stretches, the introns. Think of them as blank pages or filler text in the manual. When the gene is first copied into RNA, it includes all the exons and introns. Then comes the splicing process, where enzymes act like scissors and glue, cutting out the introns and sticking the exons together in the correct order to make the final instruction manual mRNA ready for protein production. But here's the kicker. What if the splicing scissors don't follow the exact same pattern everywhere? What if in the brain they cut out intron A and exon 3, but in the liver they cut out intron B and exon 2? Suddenly, you have different proteins made from the same original gene blueprint. This is alternative splicing. It's like having one set of instructions for a chair, but being able to assemble different variations, maybe a rocking chair version using exons 1 and 2, a lounge chair using exons 1 and 3, or even just a simple stool using only exon 1. This completely blows up the one gene, one protein idea. In humans, this isn't rare. It's the norm for about 90% of our genes. And remember those transcription factors we talked about? They can often influence which splicing pattern is used in a particular cell type. It's another layer of complexity, another loop. Even the enzymes doing the splicing are proteins, coded by their own genes. It's a tangled web. Now for perhaps the most radical challenge to genetic stability, transposable elements or jumping genes. Back in the 1940s, Barbara McClintock, studying corn kernels, noticed mutation patterns that made no sense under existing theories. Her only explanation, that chunks of DNA were copying themselves and inserting into new spots in the genome. She called them transposons. People laughed. They called her crazy. She was largely ignored for years, working in isolation. But then, with the rise of molecular biology in the 70s, she was proven right. Transposons exist. Now, most of the time, these jumps produce gibberish, like taking a sentence and randomly shuffling words into nonsense. The example given is hilarious. The fertilized egg is implanted in the uterus, becomes the fertilized egg terrace is implanted in the uterus. Utterly useless. However, sometimes, rarely, a jump lands in just the right place to create something novel, like changing egg to eggplant in that sentence. Plants actually use this stress-induced shuffling as a desperate gamble for survival under drought. And our immune system uses it cleverly to generate the incredible diversity needed to fight off constantly evolving viruses. But here's the really mind-bending part. Transposons happen in the brain. During neurogenesis, when stem cells become neurons, these random DNA jumps occur. So the neurons you end up with aren't all identical copies of the DNA you inherited. Your brain is a mosaic, a patchwork quilt of neurons, each potentially carrying slightly different DNA sequences due to these transpositional events. It's like every neuron has its own unique little scribble, or typo in the original script. Even fruit flies do this in their memory-forming neurons. It suggests that even our fundamental thoughts and memories might arise from a brain whose very building blocks are genetically unique. And finally, let's not forget chance. At the molecular level, things are constantly bumping into each other randomly, Brownian motion. How quickly a TF finds its target, how efficiently splicing happens, how fast synthesis occurs. All these processes are subject to random fluctuations. So even if the rules are set, the execution involves a lot of luck. So, let's recap the main points from this deep dive into the nitty-gritty of genetics. First, genes aren't little robots running around independently. They're heavily regulated by everything happening around them, the whole environment, inside and out. Second, most of our DNA isn't just code for proteins. It's involved in turning other genes on and off. Evolution, it seems, is mostly about tweaking these regulatory switches, not just inventing new protein blueprints. Third, epigenetics shows how experiences can lock genes into certain states, sometimes permanently, and even pass these effects down. Fourth, thanks to transposons, the very neurons in our brains might carry unique genetic variations. The big takeaway: genes don't determine much in isolation. They operate within a complex web of regulation, chance, and environmental influence. This theme continues as we look at how genes supposedly affect behavior. Okay, shifting gears slightly. Before we knew anything about promoters or splicing, scientists started looking at behavior genetics from the top down. The basic idea is simple. Look at families. If everyone in a family does something, maybe it's genetic. Sounds reasonable, but it's flawed because families also share environments. So the next step was comparing closer relatives versus distant ones. If a trait is more common among siblings than cousins, it must be genetic, right? Again, flawed because siblings share more environment too. Researchers got smarter, using tricks like comparing someone to their biological aunt versus their uncle. The aunt shares genes plus environment. The uncle shares only environment. Any extra similarity with the aunt points to genetics. Clever but still problematic. We need better tools to really untangle genes from environment. Then came the twin revolution. Identical twins are like two clones, sharing 100% of their genes. Fraternal twins, DZ, are like regular siblings, sharing about 50%. Here's the genius idea. Compare MZ twins raised together with DZ twins raised together. Both pairs share the same age, the same home environment, the same prenatal environment. The only difference is the amount of shared genes. If a trait appears in one twin, how often does it appear in the other? If MZ twins show higher concordance, both have it, than DZ twins, the difference must be due to the extra 50% of shared genes in the MZ pair. This sounds great, but it hinges on a huge assumption that MZ and DZ twins are treated equally by their parents and peers after birth. Is that really true? Another powerful tool emerged in the 1960s, adoption studies. This is beautifully simple. Find someone adopted as an infant. They share genes with their birth parents, but grew up in the same environment as their adoptive parents. Compare the adopted person to both sets of parents. If they resemble their biological parent more on a trait, it suggests genetics played a role. If they resemble their adoptive parent more, it suggests environment played a role. It's the classic cross-fostering experiment done in humans. This approach was crucial in early research, helping establish that schizophrenia has a significant genetic component. And then the holy grail of behavior genetics, identical twins separated at birth. This is the ultimate natural experiment. You have two individuals with identical genomes, but raised in completely different environments. What similarities remain? Thomas Bouchard and his team found dozens of these pairs. The results were stunning. Despite being raised apart, reunited twins often show 惊人的相似的个性、兴趣爱好，甚至是一些怪癖，比如冲马桶的习惯。One famous pair, Jack Yufei and Oscar Storr, were separated at 6 months. One became an observant Jew in Trinidad and Israel, the other a Hitler Youth member in Nazi Germany. Yet, they shared numerous traits, including that toilet flushing habit. Studies consistently showed major genetic influences on everything from IQ and personality to mental illness, religiosity, and even attitudes towards authority. Same genes, different lives, yet remarkable similarities persisted. So, these twin and adoption studies generated tons of data, often showing high heritability scores for various behaviors. But here's a crucial point often missed in headlines screaming, "Scientists find gene for X." High heritability doesn't mean there's a single gene responsible for the behavior itself. It means genetics explains a large portion of the variation between individuals in that population. Often, the genetic influence works through incredibly indirect pathways. Want to understand why some people vote and others don't? It turns out genetics might influence personality traits like optimism or locus of control, which then affects voting behavior. Or consider self-confidence. Some studies suggest genetics influences height, and taller people tend to be treated better and feel more confident. See? It's rarely a direct gene to behavior link. It's more like gene to personality trait to behavior, or gene to physical trait to social treatment to behavior. Much more complicated than a simple switch. Of course, these elegant studies aren't without serious critics. The biggest issue is the equal environment assumption for twins. Do parents really treat identical twins identically while treating fraternal twins differently? Probably not entirely, but maybe enough to skew results. If MZ twins are treated more similarly, their higher similarity might be environmental, not genetic. Then there's the prenatal environment. Most identical twins share a placenta, meaning they bathe in the same maternal hormones and nutrients in the womb, something fraternal twins usually don't experience. This shared fetal environment could explain some similarities. Adoption studies also have confounds. What about the 9 months in the womb with mom? What about epigenetic marks passed via sperm or egg? What about selective placement? Agencies often try to match adoptive families with the child's racial or ethnic background, which might mean they also share some genes. And finally, adoptive parents often come from a different socioeconomic stratum than biological parents, creating a less diverse range of environments, which can make genetic effects look stronger than they are. These critiques are real, though efforts to control for them often suggest the inflation is relatively small, especially for psychiatric disorders studied. Now, let's tackle a concept that trips up almost everyone: heritability. When behavior genetics studies report a heritability score, say 50% for intelligence, what does that mean? It's easy to misinterpret. Heritability is a statistical measure for a specific population in a specific environment. It tells you what percentage of the differences between individuals in that group can be explained by genetic differences. It's not about whether the trait exists in the species. That's inheritance. Humans having 5 fingers is strongly inherited, but the heritability of finger number is low because most variation extra fingers syndactyly comes from accidents, not genes. Conversely, wearing earrings in 1950s America wasn't strongly inherited. Men rarely wore them, but the heritability was probably very high gender linked to X chromosome because it perfectly divided the population along sex lines. The problem is, people usually care more about why you are different from your neighbor, heritability, than why humans are different from apes, inheritance. And research often inflates these heritability scores. Why do heritability scores get inflated? Consider our imaginary plant. Gene 3027 has versions A, B, C yielding 1, 2, 3 inches tall. Studied in the desert, height is perfectly heritable, 100%. Studied in the rainforest, it's also perfectly heritable, 100%. But combine the data. Knowing the gene version tells you almost nothing compared to knowing if it grew in the desert or the rainforest. Heritability plummets. This is critical. Heritability scores only tell you how much genes explain variation in the specific environments where the study was done. Study a trait in only one environment, and you automatically inflate the apparent importance of genes because you can't see how it behaves elsewhere. The more diverse the environments you study, the lower the overall heritability tends to be. Controlled lab experiments, while cleaner, artificially inflate heritability because they prevent discovering these hidden environmental factors. Bouchard himself warned that conclusions apply only to populations exposed to similar ranges of environments as those studied. This leads us to the absolute cornerstone of modern behavioral genetics, gene-environment interactions, or GRICs. This is where things get really interesting. An interaction occurs when the effect of a gene depends on the environment, and the effect of the environment depends on the gene version. It's a two-way street. How do you recognize it? Ask yourself, what does this gene do? If the answer is it depends on the environment, then ask, what does this environment do? If the answer is it depends on the gene version, bingo. You've got an interaction. Look at our plant again. If gene versions always yield 1, 2, 3 inches regardless of environment, that's no interaction. But if version A yields 1 inch in environment X, but 3 inches in environment Y, while version B does the opposite, 3 inches X, 1 inch in Y, that's a classic interaction. The gene's effect flips depending on the environment. This means talking about a gene's effect in isolation is basically meaningless. Its effect is always relative to the context. These interactions aren't just theoretical curiosities, they're everywhere. Take PKU. The gene mutation is harmful only if you eat normal food containing phenylalanine. Diet interacts with the gene. Depression risk linked to a serotonin transporter gene variant? Only if you also experience childhood trauma. The gene interacts with the environment. A gene involved in fat metabolism linked to higher IQ. Only if you were breastfed. Interaction again. And here's the kicker from a 1999 study. Researchers took genetically identical mouse strains and tested them identically across 3 different labs. Oregon, Albany, Alberta. Same mice, same tests, same conditions. Except for subtle, unknown differences between the labs. And guess what? For some genes, the results varied massively. One strain's response to cocaine was 667 centimeters movement increase in Oregon, 701 centimeters in Albany, but over 5,000 centimeters in Alberta. Identical triplets pole vaulting wildly different distances, despite identical training. This screams GRAC. Even in super-controlled settings, tiny environmental differences can dramatically alter what a gene does. What does this mean practically? Donald Hebb nailed it. Asking if nature or nurture matters more for a trait is like asking if length or width matters more for the area of a rectangle. Both are essential components. Consider cognitive development. Studies show the heritability of IQ is around 70% in kids from high socioeconomic status families, but only around 10% in low SES kids. Poverty, it seems, can stifle genetic potential. Similarly, the heritability of alcohol use is lower among religious individuals compared to non-religious ones. If your environment strongly discourages drinking, your genes might have less room to play out. These examples highlight the power of context. Genes aren't operating in a vacuum. Their influence is profoundly shaped by the world around them. So, we've established that genes interact with the environment. Now let's try to connect the dots using molecular genetics. We take the insights from twin and adoption studies, genes matter, and use tools to find the actual genes involved. One approach is candidate genes. Pick genes already known to be biologically relevant. If you're interested in aggression and serotonin, look at the serotonin system genes. Use knockout mice, where you delete the gene, or transgenic mice, with extra copies, to see what happens behaviorally. Then, check if variants of that gene in humans correlate with aggression. This marriage of approaches has yielded fascinating, but also messy, results. Many findings don't replicate easily, the effects are often tiny, a gene might contribute but not dominate, and the most important findings almost always involve those pesky gene-environment interactions. Let's look at the serotonin system, which has gotten a lot of attention mostly for the wrong reasons. Pharmacology and animal studies strongly suggest that low serotonin levels foster impulsive aggression. Drugs that lower serotonin increase aggression, drugs that raise it decrease it. So, intuitively, you'd predict that genes leading to low serotonin should correlate with aggression. Genes that make less serotonin, low activity TH, break it down faster, high activity MAOA, or scoop it up quickly, high activity 5-HTT transporter, or have receptors less sensitive to it. But here's the twist. The genetic data often goes the opposite way. The MAOA gene, when mutated to be inactive, no breakdown, so high serotonin, is famously linked to aggression in the Dutch family and hyperaggressive mice. But studies on common variants of MAOA, the low-activity type, also show links to aggression. Same goes for the 5-HTT transporter gene. The variant that leads to less reuptake, higher serotonin, is associated with more aggression. This flies in the face of the simple drug-induced picture. Why the contradiction? Maybe the body compensates chronically for lifelong genetic differences differently than it responds to a short-term drug dose. Maybe the developmental effects matter? Whatever the reason, the simple story is broken. This MAOA story blew up into the infamous warrior gene narrative. The idea that having this low-activity variant meant you were doomed to be violent. It even got cited in court to argue for reduced sentences for murderers. Responsible scientists recoiled in horror. The effects are tiny, the gene does other things, breaks down norepinephrine too, and the original Dutch family wasn't just violent. They had arsonists and exhibitionists. But the biggest blow to the warrior gene idea comes from gene-environment interaction. The landmark Caspi study followed people from birth. Did the MAO variant predict antisocial behavior? No, but did it predict it when combined with severe childhood abuse? Yes. Having the low-activity variant tripled the risk of antisocial behavior, but only if you'd been abused as a child. Without abuse, the variant was irrelevant. Warrior gene? More like warrior environment interacts with warrior gene. Context is everything. Moving on to dopamine, the reward molecule. Variants affecting dopamine signaling, receptors breakdown reuptake, are often linked to sensation-seeking, risk-taking, impulsivity. The DRD4 gene coding for a dopamine receptor is supervariable. The 7R variant is linked to novelty-seeking, ADHD, impulsivity. But guess what? It's also linked to less generosity, unless securely attached, less interest in pro-social causes, unless reminded of religion, and worse patience, unless raised wealthy. Again, context. COMT is the enzyme that breaks down dopamine. The efficient version is linked to higher extroversion and aggression. And guess what? Anger traits, but only if coupled with childhood sexual abuse. Dad is the transporter that reuptakes dopamine. Less efficient versions, more dopamine, are linked to preferring happy faces and positive parenting. But the picture is messy. Effect sizes are tiny. The roles of COMT and DAT might overlap or differ of different brain regions. And COMT messes with norepinephrine too. It's complicated. Next up, the social hormones oxytocin and vasopressin. We know they boost prosociality, trust, empathy, bonding, but also just general social information gathering, and can sometimes make us hostile towards outsiders. Genetics seems to play a role. Variants linked to higher levels of these hormones or their receptors generally correlate with better relationships, parenting, empathy. But then there are conflicting studies linking other variants to extreme aggression or social disconnection in kids. The problem? We don't know if these variants boost or dampen the signaling. And of course, GREX is rampant here. One oxytocin receptor variant predicts less sensitive mothering, but only if the mother had a tough childhood. Another predicts aggression, but only when people are drunk. Yet another predicts seeking support during stress, but only among Americans, not Koreans. Culture matters. It's a tangled web of genes, hormones, and context. Finally, let's touch on steroid hormone genes. There's no testosterone gene per se, but genes for enzymes that make it, convert it to estrogen, and the androgen receptor. Receptor variants linked to sensitivity to testosterone are intriguingly linked to violent crime in criminals and masculinization of the cortex in boys. And yes, GREX again, high testosterone plus a potent receptor variant predicts aggressive mood. Estrogen receptor variants show sex differences, linked to anxiety in women and antisocial behavior in men. Mouse studies show the receptor's presence affects female aggression based on litter size. Adding genetic information about hormone systems gives a slight edge in predicting behavior, but it's still modest compared to just knowing hormone levels. So, wrapping up this section, the picture is clear. Genes are incredibly complex, regulated, modular, and even mutable through transposons. Epigenetics shows environment can lock things in for life or generations. Heritability scores are fragile measures tied to specific contexts. And gene-environment interactions are everywhere, making it impossible to talk about what a gene does without talking about the environment. When we look at specific genes, the links to behavior are often subtle, indirect, and heavily dependent on context. The simple idea of a warrior gene is a myth. Understanding the relationship between genes and behavior requires embracing this complexity, this interplay between nature and nurture.